So it's a pleasure to be here with you today and to be able to share a little bit of some of the research that's going on in my lab. This is only one facet, actually. So I also have some other research related to traumatic brain injury, specifically related to how that affects the eye, as well as looking at myopia or nearsightedness. But today, I'm going to talk to you about how to improve detection and treatment of diabetic retinopathy. Maybe. So, okay. Let me just start this again. All right. So I'm sure that most of you are aware that diabetes is a chronic disease of high blood glucose levels. And there's two main types. There's type 1, where you have little or no insulin that is excreted from the pancreas, or you have type 2, which is poor regulation of insulin. And in type 2, obesity also seems to be a major risk factor. So the prevalence of diabetes has been increasing at alarming rates. And I'm going to show you some CDC heat maps that show you diabetes diagnosis across the US. And this is from 2004 to 2009. So the dates are a little, in the past, but it's still very relevant. And I think you're going to be impressed at seeing this. So here you see the heat map, and you notice the percent. So we go from 0 to 6, and then the darkest red here is over 11.2. Now, of course, we're down here in the south, so you can keep your eye on that, but try to just pay attention to the whole map. Okay, 2004, 2005, 2006, 2007, 2008, 2009. All right, so the increase in the number of people that have been diagnosed with diabetes has just dramatically increased in the most recent years. And it's a little bit scary. It's particularly scary when we think about how many diabetic complications there are associated with diabetes. And I'm particularly interested in diabetic retinopathy, so how diabetes affects the retina. So this slide shows us some very common eye diseases, and I'm particularly interested in diabetic retinopathy here. So the current number of cases in the US is about 7.6 million, and this is a little graphic from the National Eye Institute, and you can see the percentage of the wedge here in the iris of the increase that they expect by 2050 of each one of these diseases. So this is a major problem. Diabetic retinopathy is the leading cause of retinal disease and vision loss in the non-elderly, like people that are in their working ages. And about 30% of people that have diabetes have diabetic retinopathy. That's about 4.2 million adults that actually have diabetic retinopathy. Now, there's a lot of cost associated with this, and it's estimated about 500 million annually. So the CDC has a couple prevention tips. The first is just good control of blood sugar and blood pressure levels, and then early detection and treatment of eye disease. And that's the one that I am most interested in, is how can we detect this disease earlier, and then how can we treat it? And so my talk today in my lab in particular is very focused on these two things. Early detection, I'm going to talk to you about ways we can have early detection, and treatment for key elements in maintaining vision. Now, when we think about detection of retinal disease, there's some key elements that we're most interested in because we want something that we can use as a screening tool. So ideally, this would be something that's non-invasive, that would be very easy to perform, such that it could be used in any kind of outpatient clinic or maybe even in somebody's home. We would also want to have a very high throughput, and it would be very inexpensive. Now, let me tell you a little bit more about diabetic retinopathy and how it's currently diagnosed. So it's clinically recognized as a vascular disease. And so patients that have diabetes are told to go and get an annual eye exam where the doctor will dilate their eyes and they'll look in with a fundus camera to look at the blood vessels. And when they look in, they might see something like this, some hemorrhages indicating bleeding, some cotton wool spots, or even some neovascularization that happens in more late-stage disease. Now, often the patients may not really notice that they have these earliest signs of vascular defects. And so it's really this detection with the fundus camera that allows this to be noticed. And there's also some other things that are happening probably along with this at the microscopic level. We have the loss of these pericytes that are in the blood vessels, and we have the formation of microaneurysms that are even smaller than what we can see in the fundus exam. So this is probably mid to late stage disease. So when the ophthalmologist is able to actually see these structural changes, the disease has been there for quite some time. And at this point, vision loss is irreversible. Okay? They are definitely going to have vision loss. So let me tell you about a study that has been done to kind of emphasize the fact that having early detection is really important. And this is a study that was done in diabetic rats in Renu Kaluru's lab. She's up at Wayne State. And she took these rats and she put them into different groups. So some of them had poor glycemic control, 12 months of poor glycemic control. Some of them had 6 months of poor, followed by 6 months of good. And then another group had 12 months of good glycemic control. And she's interested in looking at mitochondrial DNA. And so she looked at the amount of mitochondrial DNA here. And you can see the normal bar. And you can see that if you have poor glycemic control, that you have a decrease in mitochondrial DNA. And if you have good glycemic control over the 12 months, you have basically normal mitochondrial DNA. But what's important and what's clinically relevant is that middle group, where somebody has poor glycemic control for 6 months. That may be because the patient really doesn't know that they're diabetic yet. And then they get put on insulin and they have good glycemic control. But look, it didn't actually make a difference to that mitochondrial DNA. All right, so we already have damage that's there that can't be reversed. So when we think about disease, when we think about the processes that happen to disease, you might be able to think about it happening in these stages, where the first stage is kind of adaptive changes, things that are happening all the time in our body that we don't consider pathology. They could be oxidative stress or even some neuronal dysfunction or maybe even some mitochondrial DNA damage. But these are things that our body can repair quite easily. And then we move into another stage, which we could consider early pathology, which also has oxidative stress, neuronal dysfunction, mitochondrial DNA damage, and vascular dysfunction. But these would be reversible changes, things that we could actually go in and change. So there's some window where we have that. And then there's end-stage pathology, where these things lead to vascular leakage, neovascularization, or blindness. And these are the irreversible damage. And this is really where DR is currently being diagnosed. Now, ideally, we would like to be able to take and go from the adaptive, have this early detection right here in early pathology, and be able to just have this maintain so it doesn't ever advance to end stage or even fall back down into the adaptive zone. That would be ideal. So my lab is asking whether it's possible to detect diabetic retinopathy prior to vascular structural changes. And to do this, we are looking at various aspects of function. We're looking at retinal function itself, visual function. We're also doing some in vivo imaging with some special markers that I'm going to tell you about. And we're looking at neurovascular coupling. Now, one of the things that I need to tell you as we go forward is when do vascular changes actually happen in a rat? Because that's going to be important if I'm telling you that I want to detect something earlier than those vascular changes. So in the STZ rat model that we're going to use, they show vascular changes after about 4 to 6 months, with the first signs being that pericyte dropout, and you'll get some leakage. Now, interestingly, the rats don't ever develop neovascularization like humans do, but they still have some of those early structural vascular changes. So first, I'm going to start with talking about retinal function. So if we want to measure retinal function in the eye, we do so using an electroretinogram, or an ERG. And this is very similar to an EKG, and it's a full field electrical potential of the response of those retinal neurons to a flash of light. And we record this by placing an active recording electrode on the surface of the cornea. Okay, so it's just laid across, the eye is numbed with just a numbing drop. And then we get this very characteristic waveform here. And this is the full waveform where we have this negative going wave called an A wave, a positive wave called a B wave, and then we have these little wavelets called oscillatory potentials that we can filter out, and you can see them here, and we number each one of those. All right, so this is recording that's done clinically. We can also do it in the lab in animals. So one of the really important things about this waveform is that we know exactly what cell types make up each different wave. And so here I'm showing you now a family of waves. This is a graded response. So this is a dim flash to a very bright flash. And you can see that in these family of waves, the amplitude is getting larger and it's also getting faster. And on this side, I'm showing you a nice immunohistochemical micrograph of the retina. And it's a beautiful structure, all these very nice layers from the ganglion cells all the way to the photoreceptors. Now this big positive wave, this B wave, is actually generated by these green cells, these on bipolar cells that are in the retina. And this negative going A wave here, it's actually generated by these photoreceptor cells. And then these little oscillatory potentials, these wavelets, they're generated by amacrine cells in the inner retina. So most of them being in this area and a few being in the ganglion cell layer. Now there's another important aspect about the ERG that you need to keep in mind. And that is that we can actually probe different pathways within the retina depending on what flash intensity we use. So if the flash intensity is really dim, then we are only stimulating rod pathways, okay? Because the rods are very sensitive to dim light. But as we get brighter and brighter, we begin to also stimulate the cones. And so in these brighter flashes, we're stimulating a mixed response of both rod and cone photoreceptors. All right, so when we look into the literature to look at retinal dysfunction with diabetes, we find that there's quite a bit of evidence that the ERG changes. And in fact, back in the 1960s were the first reports of an ERG delay using a very bright flash stimuli. And I'm giving you some example waveforms that kind of show you what this looks like. So this is an individual with no retinopathy. These are these little oscillatory potentials. And you can see that these are different stages of retinopathy where it's progressing to worse and worse stages, more advanced, till we get here to this proliferative retinopathy with hemorrhages. So this would be neovascularization is actually occurring. Now, I've drawn a vertical line here so that you can visually look at the first oscillatory potential. And I hope that you can see that it's delayed in each one of these progressive stages. In addition, look at how these oscillatory potentials are getting smaller and smaller with disease until you get to that end stage and they're really not recordable anymore. So based on looking at the literature, we asked, well, what are the earliest functional delays that we can actually see in diabetic retinopathy? And in order to do that, we looked at a diabetic rat. So this is using STZ, streptozotocin, which is a toxin that'll kill the beta cells in the pancreas. And this induces hyperglycemia. We're using log Evans rats, and this is considered a model of type 1 diabetes. Okay? We consider the animals to be diabetic when the hyperglycemia is greater than 250 mg per deciliter. And to some of the rats, we give them an insulin pellet, not to reverse their hyperglycemia, but just to prevent weight loss. Okay? And that keeps them a little bit healthier over the course of the experiment. So their hyperglycemic state still remains. Then we're just doing this functional assessment of dark-adapted ERGs. Okay? And again, remember that in the STZ rat, vascular changes won't occur until 4 to 6 months. So we then went and we did repeated measurements over the course of these diabetes. And you can see in this experiment, we have control animals, and then we're looking at 4, 8, and 12 weeks post-STZ.
Blue jeans.
All right. So On this side of the slide, I am showing you the rod-only stimuli. So this is in response to a dim flash, okay? So this is really only probing those rod pathways. And on this side, I'm showing you a mixed rod and cone stimuli, something that's much brighter. So what I want you to notice with the dim delayed, and it's progressively delayed as we go out to 12 weeks post-STZ. But when we use a bright flash, which is what's usually used clinically, you can see that we don't actually see this early change. We only see a change at this 12-week post-STZ mark. Okay? So this seems to indicate that rod-driven intraretinal responses are affected very early in diabetic retinopathy. So let me show you this a different way. This is another set of rats, another cohort. These we followed for much longer, from 7 to 29 weeks. And you can see the dim flash response or the bright flash response. Again, you can see the progression of the delay with the dim, but in the bright we don't see any loss. And I think it's really interesting when you look at this as an intensity response function. So here I'm plotting implicit time over the flash stimuli. And the control, 95% confidence interval, is in the dashed line. And you can clearly see that the diabetic animals begin to show some delay at the 7-week time point. And by 14 weeks, this is clearly significant at the dimmest flashes. And then you can see that it becomes more significant, and the number of flashes that are involved continue to get to the brighter and brighter levels as we progress out. So this has hallmarks of being able to be useful for some sort of screening tool. And as I mentioned, when we look at clinical ERGs, we don't actually look at oscillatory potentials under the dim flash. So when we do a clinical ERG, we usually try to minimize the number of flash that we're going to use in general. So it's a little uncomfortable for most people to have an electrode on their eye. So you're always balancing that discomfort with how much information you can get from the test. So typically in the clinic, we do one scotopic dim, one scotopic bright, and then we do the standard oscillatory potential analysis just on this bright flash. So in order to see whether or not the results that we were getting in the animals was also going to be visible in the human patients, we decided to run some ERGs using a couple other test flashes. So now this is 2 other test flashes that we're going to add in to this clinical protocol. And we had 3 different patient groups. We had control healthy individuals. We had patients that were diabetic but had not yet been diagnosed with retinopathy. And then we had patients that did in fact have retinopathy. So let me show you what their ERGs look like. These are those same oscillatory potential waveforms I've been showing you. And this is the ICEV, this dim flash. And you can see that the oscillatory potentials are very hard to see. And in fact, in the patient with diabetic retinopathy, there's nothing there. But in our test flash 1, you can see that we have some nice waves that we can measure. And we also see that there's a little bit of a delay that we can see. So let me show you that quantitatively. So here I'm showing you, again, an intensity response function. Here's our time. Here's our flash stimuli. The 2 flashes with the orange arrows are our 2 test flashes. So I'm showing you that dim one that really wasn't very visible. And look, with these patients that are diabetic but have not yet been diagnosed with retinopathy, we clearly see a delay here that's significant, indicating that they have some sort of rod function, rod dysfunction that's occurring. Now, when we look at the patients that also have retinopathy, they also have intensities. But I'm not so interested in that, because these patients, we can already look in their eyes and we can see that they have retinopathy because they have structural changes. Okay? So it's really this flash intensity that's important. It might be an early screening tool. All right, so let me move on now to look at another type of function we could measure, which would be visual function. And to do this, we are going to use a reflexive behavior in animals called the optokinetic response, or optokinetic tracking. And what we do is we put monitors that create a little box, and then the animal sits on this stand. And what they see on these computer monitors is spatial frequency gratings. So black and white stripes that are rotating around the animal. When they can see those stripes rotating, they actually will do a reflexive head turn in the direction of the moving stripes. So the experimenter will observe this from a camera that's mounted at the top of the box, and then you simply tell the program whether or not the animal's seen the stripe or not. And there's a stepwise paradigm that happens where you can find the threshold of the spatial frequency threshold, the thinnest stripe that they can see, very similar to visual acuity measurements in humans, or contrast sensitivity, graying the stripes until there is gray till you find that threshold. So these are very similar to what we can do clinically. And when we look at these measurements in the diabetic rats, we see that we have a very clear decrease in function. So here we have the visual acuity or the spatial frequency threshold. The red is our diabetic animals. And you can see that they quickly decline in this visual function measure where the controls are pretty stable. And if we look at contrast sensitivity, we also see a decline. So notice that it's significant here at about 3 weeks. And it's significant here in this cohort about 9 weeks, although we have other cohorts that show it as early as 4 weeks. So we're clearly seeing some functional changes that are happening. Now, one of the things that I think is really interesting about this test is the fact that in the clinic, we mostly use static tests. We use the eye chart that you're used to with the big E, and nothing's moving. These are moving gratings, which are actually going to stimulate different retinal pathways. And we have special retinal ganglion cells in the eye that are specific to motion detection. So it may be telling us that there's actually different susceptibility to different retinal cell types, and that may be important. Yes, Lena?
Is this test also sensitive to the dimness or brightness of the light? Or?
It can be. We can run the test under scotopic or like dark adapted conditions with very dim stimuli. So we could try to probe broad pathways versus cone. We've done a little bit of that. We haven't really seen. We definitely get a deficit when we do that, but it's not clear that it's worth the effort, to tell you the truth. So. Okay, so moving right along, I'm now going to tell you about our in vivo imaging that we're doing. And first, I need to tell you a little bit about the mechanisms that underlie diabetic retinopathy. And so this shows in a very complicated way that there are lots of different pathways that are implicated. And so we have all of these pathways along the left-hand side here. And notice that they're all basically causing oxidative stress. And in fact, all these different arrows, whether we go to mitochondrial dysfunction, we have this increase in oxidative stress that's going to finally lead to retinopathy. So this is a really nice target then to be able to look at whether or not we have the disease process actually occurring. And so in order to try to non-invasively image oxidative stress, we've been using these molecules called hydrocyanines. So this is a fluorescent probe that can detect reactive oxygen species. It is non-fluorescing, and then when it binds to reactive oxygen species, it will fluoresce, and it becomes membrane impermeable. Now, this was first developed by Niren Murthy when he was here at Georgia Tech, and there's some other labs that have been using it here for cell culture or ex vivo work, and in vivo for muscle. Or here in this example, this is a LPS model, so they've injected the LPS into the abdominal cavity, and you can see here's the saline injected animal, and then with the dye, and here's the LPS animal, and you can see this increase in the fluorescence that's happening in the abdominal wall. So when I saw this particular marker, this was very exciting for two reasons. One is because the eye is extremely accessible for imaging. It's marvelous because I can non-invasively look into the eye and I can see the retina. So anything that would be a marker is very easily seen. The other thing that's really nice about these hydrocyanines is that they have different flavors, if you will, different wavelengths of emission and excitation. And some of them match what is currently used in ophthalmology clinics, particularly ICG. And so that is something that's already used routinely, which means that in the ophthalmic equipment, those filters are already built in. So this would be very easy to translate into the clinic if it worked. So it seems to be non-toxic in animal models, highly sensitive, stable, and it binds very rapidly. So let me show you some of the results that we've gotten from this. This is a validation experiment, and we're using a scanning laser ophthalmoscope to actually visualize the fluorescence. And how we've done this experiment is we've induced basically an injury to the retina by using toxic light. So if we expose rodents to very bright light, like 10,000 lux, the animals will develop a photoreceptor degeneration, and that's going to produce some oxidative stress. And so we either gave them no light exposure or 1, 2, 4, or 6 hours of this very bright light exposure. And when we did this, you can see that as we increase the light exposure, we have more and more fluorescence that's actually visible in the retina. And this middle part here is the optic nerve that you see. In this panel, we're actually just reverse the colors. I think it's easier to see when the colors are reversed. And so you can see a little bit of the fluorescence here that's already showing up and how it gets darker. And also the area gets larger. So I'm plotting the quantification using mean fluorescence, as well as the area of hyperfluorescence, and that is also pretty linear. And then we wanted to see, well, where is this occurring? Is it binding at the right spot? And when we look at a cross-section of the retina, this is a control retina showing no fluorescence. And this is an animal that has had light damage. And you can see these inner segments here are lighting up. And those are where the mitochondria are in the photoreceptors. It's exactly where we would expect to see that oxidative stress occurring. So it appears that this is binding oxidative stress in the photoreceptors as we predict. And so now we're just getting ready to start experiments really looking at models like the diabetic retinopathy model and seeing if this is going to be useful for detecting early oxidative stress in that model. All right, now the last type of detection that I'd like to talk to you about is neurovascular coupling. And so I've already told you that we certainly have vascular changes that are happening in diabetic retinopathy. But it's also been reported that there's some problems with neurovascular coupling. And of course, neurons in the vascular, they're very tightly linked, right? So it makes sense that we'd have to look at both. Matter of fact, even all the stuff I've shown you already, which I could declare is all neuronal, you could argue, well, if the vasculature or the neurovascular coupling is defective, that would affect that function, and I would totally have to agree. So we wanted to look at this, and you can see here in this little schematic that we have normal retinal function where we have some sort of retinal stimulation with light, and that produces ATP that is going to stimulate in the Mueller cell some intercellular calcium increase, and that is going to stimulate these arachidonic acids. And nitric oxide also regulates them. And so they have two different flavors, the CYP4A or CYP2C11, and the arachidonic acid metabolites of interest are 20-HEAT and EET, which are going to cause vasoconstriction or vasodilation of the arterioles. Now, what happens in the diabetic retina? Well, we don't really know, but what we hypothesize is that we have processes happening like that oxidative stress, inflammation, that's going to increase iNOS, that might increase retinal nitric oxide levels, and that is going to affect this CYP4A and CYP2C11. But you can see that it inhibits CYP2C11 greater than it does the CYP4A. And that leads to an increase in the 20-HEAT to EET ratio. So these now are not well balanced, and that leads to decreased functional hyperemia. And the functional hyperemia being a response of the retina to some sort of flickering light, okay? Now, how do we measure that in the rat eye? Well, we take a rat and we flicker some green light at the eye. At the same time, we can image that eye with a scanning laser ophthalmoscope by placing a prism mirror in the path of the light. And when we look into the eye, this is what we see. So these are the different vessels. You can see veins and arteries have been labeled here. And as we flicker the light, I should also mention we add a a dye into the blood vessels called ICG. And so this indocyanine green makes these blood vessels very visible. And then we're simply going to measure the diameter of that blood vessel in response to the flashing light. So when we do that, we can see some relative changes here. So here's the arterial vessel caliber across time. This is when the light stimulation turns on. You can see the control is increasing and then decreasing. The diabetic also increases and then gets down to baseline, but the diabetic doesn't have as much change. That's much more visible when we quantify it here. So this is percent vasodilation. You can see we get about almost 12% in the controls and more like 7% in the diabetics. And we see a similar type of response in the venules, but it doesn't quite reach significance. All right, so we seem to have some sort of dysfunction in the neurovascular coupling, and other people have also published that. And note that this is very early. This is at 2 weeks post-STZ. So this is earlier than any of the other functional changes that I've shown you thus far. So we now wanted to say, well, okay, if we have this happening, is there a way that we could treat this? And to look at treatment, we decided to apply this drug, AUDA, which is going to increase EET levels by targeting this upstream inhibitor, epoxide hydrolase. Okay? So what happens when we do that? Well, first we looked at the functional hyperemia response at 2 weeks again. And you can see here our control animals and the diabetics that have just vehicle, and of course they have a deficit. But when we give the diabetic animals AUDA, we're able to restore their neurovascular coupling. That functional hyperemia response is normal. In fact, it's even a little above normal. And we see the same thing in the venules as well. Now, that is great that we would be able to see that functional response to recover, but does that really matter to a patient? Is that really going to affect their visual function? So we also looked at visual function in these rats. So again, using our optokinetic tracking, here's our spatial frequency thresholds. You can see our diabetic animals with the vehicle are having a decline, but when we treat with AUDA, we can see that we're able to prevent that initial decline at 4 weeks. By 6 weeks, we still get some decline. So this, we just were able to delay the drop here. But in contrast sensitivity, you can see that we were actually able to delay it at least out to 6 weeks. And so that looks pretty encouraging that if you were able to stop this neurovascular coupling defect, you might actually be able to preserve this early function. Yes?
These animals also show ERG deficits?
They do also show the ERG deficit as well. Yes, they do. Mm-hmm. Although they, yes, at the 4 or the 6 week time point, right? Mm-hmm. Okay, so I'd like to just spend this slide talking about, just kind of summarizing quickly what I've told you about early detection. So it appears that we have seen some neuronal deficits that occur very early in STZ rodents by 4 weeks post-STZ. And this is prior to those vascular structural changes. And that retinal and visual function measurements may provide detection methods for early DR. That's with that ERG dim flash or with optokinetic tracking. We're also investigating these in vivo fluorescent markers that may provide a method to detect ROS with the progression of DR. And I've shown you that neurovascular coupling defects are present very early at 2 weeks post-STZ, and that if we restore this balance of 20 heat and EET, we might be able to preserve function. All right, so if we're able to actually detect DR much earlier, this also opens up a new window for treatment. And so let's talk about treatments for diabetic retinopathy right now. Current treatments include anti-VEGF. And this is an injection therapy where the patient comes in every couple of weeks and actually gets an injection into their eye to stop blood vessel growth. Laser surgery, where the laser actually burns off the photoreceptors, so they're no longer needing oxygen, and that helps to help the neovascularization. You kill off the photoreceptors, you're killing off retina, or some corticosteroids. This is all mid, late stage disease, okay? And as I just said, we have early detection can help to reveal a new therapeutic window. So I'd now like to talk to you about a couple neuroprotective strategies that we are pursuing in the lab. I'm very interested in neuroprotection for this because if you're detecting this very early, this retinopathy, this early function, it's possible that a patient may take this particular drug or this form of rehab for many years, maybe 30, 40, 50 years, basically for the rest of their lives. So you want something that is very non-toxic, is going to be beneficial and easy to take. So I'm going to talk to you about dopamine, and I'm going to talk to you about exercise. So dopamine, I'm sure you're familiar with. It's important for in the brain. It's also important for visual function. And it's involved in circadian rhythms. Recently, it's been shown that specific dopamine receptors are actually responsible for very specific aspects of visual function, including spatial frequency or contrast sensitivity, which I think is really fascinating. Well, it also turns out that dopamine is decreased in STD rodent models, not only in the eye, but actually in other parts of the body as well, in the brain and in the kidneys. And we were able to replicate this result. You can see clearly that there's a drop in dopamine, as well as DOPAC, which is a dopamine metabolite in the retina of the diabetic animal. So I had an MD PhD student in the lab, Harry Ong, and he was looking into this, and he said, well, if the dopamine is decreased, well, let's just give the animals L-DOPA. This is a great translational drug. It's already used for Parkinson's disease. Let's see if it is beneficial to our rats in this early stage. So again, I'm showing you the oscillatory potentials. This is the dim flash and the bright flash. You can clearly see the delay in oscillatory potentials. With the diabetic animals that have vehicle. And when we give L-DOPA, then we can preserve that response. And we see a little bit of that same sort of trend in the bright flash animals, but of course it's not really significant. And notice this is 5 weeks post-injection, and that's for the STZ. And you can see quantitatively the delay that we're able to prevent with the L-DOPA. I also like to emphasize when we gave the L-DOPA. So we didn't give it before the animals were hyperglycemic, which is something that we often do with animals because we can kind of put everything in our favor to see whether or not a drug actually works. But we waited until these animals definitely had diabetes before we gave them the L-DOPA, okay? And I think even a better experiment is to wait even longer and see how long you can wait and still get benefit, right? And then we also look to see whether or not this helped visual function. So here I'm showing you spatial frequency and contrast sensitivity. And you can see the red line is our diabetic animals with vehicle. They clearly show a decrease like I've shown you before. And we give these diabetic animals L-DOPA, then they have significant preservation of function, whether we look at spatial frequency or we look at contrast sensitivity. So it appears that the L-DOPA is having a very positive effect. But what I find even more remarkable is that when Harry did experiments with dopamine receptor agonists, he was able to show improvements with a single injection of the agonist, okay? A very acute experiment. These animals were at 8 weeks post-STZ, and that selective difference between the D1 and the D4 receptor you can see here. So the D1 receptor was able to improve spatial frequency, and the D4 receptor was able to improve contrast sensitivity. So it isn't to the level of the controls, which is up here. I find it quite remarkable that a single dose would provide that much benefit. So just a quick summary. The dopamine precursor L-DOPA to preserve early deficits in retinal and visual function in our diabetic rodents, and a single dose of dopamine receptor agonist provides benefit. All right, now let me tell you about one other type of strategy that we're investigating, and that is physical activity or exercise. So exercise is protective and rehabilitative. I know that many of you are with this work. It seems to promote neuronal survival and axon regeneration, particularly in the hippocampus, stimulating. We know it improves cognitive function in patients that have Alzheimer's or Parkinson's or ALS. And in fact, it even enhances function in healthy aged or even young individuals. So this work, some of this work carried out in this rehab center that I'm part of at the VA, the Center for Visual and Neurocognitive Rehab. I was listening to these folks tell us about how great their research is and the benefit that the patients are getting. And I wondered whether or not exercise could have any effect on the retinal neurons specifically. So the retina is just an extension of the brain. Is it possible that it also benefited? And so we tested that. First, we tested it using treadmill exercise. And again, I'm using that light-induced retinal degeneration. So a very specific injury that kills the photoreceptors. These animals were ran 5 days a week for 60 minutes at a rate of 10 meters per minute. So this is not a race. This is not high intensity for a mouse. It's basically like a kind of a fast walk. And when we did this, you can see our controls up here. So these are animals that didn't have bright light exposure. When they had bright light and no exercise, you can see we get a pretty diminished response. And then when they have treadmill exercise, the response is preserved. This quantifies it for A wave and B wave amplitude. Here's the animals with the treadmill and the ones that did not have treadmill for A wave and B wave. Now we're not quite up to the controls, but we have significant preservation. And so this is function. This is actually photoreceptor count, so counting the number of photoreceptors. So if the animals are inactive and they get that bright light exposure, a lot of photoreceptors die, and we have about twice as many photoreceptors if we just exercise the animals. Now, this is one way to exercise. Some people, if you're familiar with exercise, you know that this is forced exercise, because in a treadmill, rodents on a treadmill have a little electrified grid at the back that keeps them on the treadmill so that they can run. And some of you may say, well, that's not very realistic to humans because we're not scared to run when we go out and run. We run on our own. It's voluntary. So we did an experiment where we gave animals just running wheels, and we looked to see whether or not that was protective. So the animals are voluntarily running every night. These animals have an inherited retinal degeneration. They're called the RD1 mice. And you can see that the inactive RD10 animals have a fast degeneration here. This is now spatial frequency thresholds. And if they have a wheel, they have significant preservation. Now, of course, the wild types have more function, but this is still very significant. And all this exercise work has been done with a collaborator, Jeff Boatwright, over in ophthalmology at Emory. Now, one of the things I'd like to point out here, too, that may be important, if you're not so familiar with visual loss, is you might think, well, we don't care if there's only this much function. We really want this function. We want to be right back to normal. And patients only are going to care if they are normal. And that's not the case. So if you go and you talk to patients that have vision loss, they would be more than happy to have some improvement. They'd be more than happy to be able to actually see faces longer, or be able to even navigate a store by themselves instead of having to rely on somebody to be able to go up and down the aisles. So there's very small improvements that make a huge difference in quality of life to the patients. And I think as scientists, we often strive for perfection, and we ignore these sorts of things where we think, oh, this isn't going to make a difference. We need to find a better drug. We need to find a better way. But actually, these sort of things could make huge improvements. And something like exercise is very non-invasive. Anybody can do it. So that is another appeal that we have. So let's see if exercise is actually beneficial to diabetic retinopathy. So here I'm showing you STZ-treated rats that have been ran on a treadmill. This is a similar sort of exercise paradigm. They're a little faster, 15 meters per minute, only 30 minutes a day, and then again 5 days a week. And we again started this exercise after the animals had confirmed hyperglycemia. And we compared them to animals that are inactive. And you can see here that our animals that are inactive are definitely getting that decrease I've showed you before. But if they have exercise, then we have significant preservation, both in spatial frequency and in contrast. All right, well, how is this actually working? What is the mechanism that's underlying this exercise effect? So one of the things that's been implicated in the hippocampus is brain-derived neurotrophic factor. And it's been shown that BDNF seems to be important for this exercise. So, sorry here. So we looked to see if exercise was in fact, if BDNF levels were normal or not in our animals that were diabetic. And I'm showing you here the retinal BDNF levels measured with an ELISA, and you can see that the BDNF levels are significantly lower in the diabetic groups. So we wanted to see if this is in fact the mechanism, and then to do that, we gave our rats a BDNF TrkB receptor antagonist called ANA12. And this was given about 2.5 hours before the animals ran on the treadmill. This kind of optimizes the pharmacokinetics of the drug. And I should also note that this is very specific to TrkB and not TrkA or TrkC receptors. And it was compared to animals that received a vehicle. So here I'm showing you the same plot of our just active and inactives with the vehicle, the orange lines, but now I've added the animals that had ANA12. And you can see that they look very similar to our diabetics that were inactive. So adding a BDNF antagonist completely blocked the protective effects, which would seem to indicate that BDNF at least is involved in this protection. Now, I would not, even though it looks pretty complete, I don't think this is the only thing going on. I think that exercise has multiple mechanisms that it's working through, but BDNF is certainly one of them. I'd also like to note that there were similar effects shown with ERG oscillatory delay, with that dim flash that I've shown you before, as well as an ERG cone flicker delay. All right, so to summarize these protective effects of exercise, it seems that the physical exercise is protective, and it's partially mediated by BDNF. And we've just plotted here the different things that we measured: spatial frequency, contrast sensitivity, oscillatory potential implicit time, or flicker implicit time, between animals that were active and inactive. And then 100% would be the control level. And so you can kind of just see the percent of improvement that the animals got with exercise. And some of them, like this flicker, which is a measure of cone function, you can see gets right back up to 100% of the control. So that's pretty exciting. And of course, there's lots of other questions that we'd like to ask about this exercise effect. We'd like to know more about the mechanism. We want to know if it's a systemic. Is the BDNF circulating through the body, or is it local? Is it being actually released from the retina specifically? Are there vascular effects? What's the duration of the effect and the timing of exercise with the disease? Can we wait much longer and still get a benefit? And then is it possible to combine the exercise with other drugs to have some sort of combination therapy, which also might be very exciting? So I'd like to start to wrap up here. This just gives you a little table to refresh your memory about what I've shown you. So we have a number of approaches that we're looking at for detection. I've told you about our current progress with those. And our future directions is what I'd like to start to think about and tell you about now. In these aspects of looking at function, we're translating these to humans. In our hydrocyanides, we're testing disease models like diabetic retinopathy. And in our neurovascular coupling, we'd like to test this more with disease progression and see how things change in neurovascular coupling. And I've told you about 3 treatments. AUDA, which was for neurovascular coupling. We need to test the long-term effects of that. And then I also told you about L-DOPA, and we're currently translating this to humans. And we're also looking for a dose response. And then we're also looking at exercise testing mechanisms, dosing, and also moving to a type 2 model. So I'd just like to take a second to tell you about our human studies that we're doing. And so this is a clinical study that we've just started in the last few weeks that is using a handheld ERG system to measure retinal function. So clinically, and even with the animals, when we do ERGs, we do it with this Gonsfeld dome here. So you can see that this is quite large and cumbersome, and we have to place an electrode onto the surface of the cornea. So this does not make a very good screening tool. But this is a handheld ERG device that was developed by a company called LKC. It has a pupil tracker in it, which means you don't have to use any dilating drops. You can also use a skin electrode, as you can see here. And for our diabetic retinopathy changes, which are only looking at the delay in the response, this setup could be really beneficial. So we're testing this right now in patients that are diabetic who don't have retinopathy. We're trying our novel flash stimuli that we're interested in and seeing whether we can detect early changes in these patients. And then we're treating these patients with L-DOPA, and we're going to see whether or not they actually have any benefit. Now, we're also doing some work with exercise. So one of the big challenges with the exercise is that patients that have different types of retinal disease do not progress very fast. And so in a normal grant cycle of 3 to 5 years, you're not going to see much of a decrease in loss of function. These things take many years to develop. And so it's a real challenge to have a neuroprotective type of strategy where you expect to just not see any loss of function, right? You're not necessarily seeing improvement, you're just hoping to stabilize them. And so in order to try to look at the benefits of exercise, we're looking for a biomarker. And so our biomarker is BDNF levels in the serum, and we want to know whether that's correlated with exercise activity. And so we're comparing BDNF levels in our animal experiments with BDNF levels in humans that are exercising to see if we can find some correlations that might be beneficial. So I'd just like to end with a couple takeaways. So DR is definitely detectable at much earlier stages than currently evaluated in the clinical setting. And if we can detect at these earlier stages, we're going to have a new therapeutic window for neuroprotective agents. I also think treating neurovascular coupling defects may prevent visual dysfunction. And there are many existing neuroprotective agents that are already out there that show much promise for DR, including dopamine and exercise. So our main goal is to preserve vision in patients that have retinal disease and to translate what we're doing in the lab to the bedside. And this is really imperative in order to treat patients that have blinding diseases. So I'd just like to acknowledge the folks that did this work. In particular, Harry Ong did a lot of the work. He's now MD, PhD. And Rachel Allen, as well as a number of undergrads that have spent time. Our collaborators over at Emory, Jeff Boatwright, Mike Yvonne, Peter Toulet, and Andy Hendrick, who's an ophthalmologist over at Emory working with us, and of course our funding sources. So thank you very much.
Thank you, Rochelle. We have a quick question. I just have one question about dopamine. Are you giving it systemically or directly to the eye because there might be other side effects?
Yeah, so we are giving it systemically, and so interestingly, there are a lot of side effects in the Parkinson's patients, but if you look at, and there's very few studies of giving L-DOPA to patients that don't have Parkinson's, but in those, they don't seem to have those side effects. So we're looking at that right now in our animal models, but we're also looking to see how low of a dose. It may be that it could be quite a bit lower than what we use in Parkinson's patients that would also help to avoid some of those side effects.
With that, I should probably let everyone go. Let's thank our speaker again.
All right.